Alright, my girls, so we left off talking about primase and how he is going to, lay down the, oh, hello, Magnolia, hello, okay, her tail. How he is going to lay down this RNA primer, which is what's shown in the red. We said RNA is ribonucleic acid. It's made with ribose instead of deoxyribose as its sugar. And for some reason that we do not know, DNA polymerase cannot add from scratch. So he must have an RNA primer. I couldn't tell you why it's an RNA primer and not a DNA primer, but that is what it is. It's an RNA primer. But DNA polymerase cannot add from scratch, and he has to add to the 3' end. Has to add on 3' end. Okay. So, with that, let's kind of talk about the 5-3 situation I mentioned in the last video over here, and the 5-3 situation I mentioned over here. So, we said that since this parent strand right here was 3' right here, that our new strand had to have its, okay, Magnolia, had to have its 5' end right here, okay? So with this 5' end being over here, and we can see that the helicase over here is going that way. And so helicase is going that way, towards the right, and the or what's not being shown is that primase also laid down a primer over here, and then DNA polymerase just started adding DNA nucleotides from there. It's not being shown, but please note that primase had to work over here as well. So in this case, each of these little nucleotides has a 5' end and a 3' end, because they're all made up of deoxyribose sugars, and these 5s and 3s, if you remember, are labeling the carbons of the sugar. And so all of these individual 5-3s, right? So there's always a 3' end that DNA polymerase is going to add a new nucleotide onto, right? So there's always an available 3' end. And so for this strand that we call the leading strand, replication is pretty smooth. DNA polymerase just steady moves along down here, no problem. DNA helicase is going that way, and so is DNA polymerase. So that's our leading strand. It's pretty smooth, pretty fast, not too difficult to replicate from there. Now we have to worry about what all of this mess is. Like, what is going on over here? So, this is our lagging strand. Our lagging strand is going to be a little bit slower than our leading strand, but overall it's not going to be significantly slower than the leading strand. And what I mean by that is DNA replication finishes at the same time for both the template strand up here and for the template strand down here. Replication is still going to finish at the same time, and I'll explain why on another slide just a little bit. So in this case, DNA helicase is still moving to the right, correct? We're still opening up in this direction over here, but in this case, our 3' end is over here, and so the 5' end is going to technically be over here, right? And so if we do the same thing that we did at the top, where we numbered the 5' and 3', right, for each of these nucleotides, it's going to be like this, right? Because we can only add 5 to 3. I'm kind of not drawing in the primes because that would take too much time. Okay, 5 to 3, 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 all the way down this strand. Okay, and we'll say this 3 over here is the last 3' end. And so what you'll notice is the 3' end is over here moving that way. Oh, okay, now my pen's just not working. Hello? Okay, a little bit of technical difficulties, but I figured it out. Okay, so we'll see that our 3' end is over here on the left, and so while the helicase is moving this way, for the lagging strand, the polymerase has to move in the opposite direction, right? The polymerase is having to move this way because he can only add on to the 3' end. If the 5' end is over here, then the 3' end is over on this side. And so the DNA polymerase is basically having to backtrack and work in the opposite direction because of this 5 to 3 nature. So let me clear this up just a little bit. Okay, so remember this is the 5' over here, and the 3' end is over here. So, let's see. It's so hard to be able to do this without actually being able to like physically show you my hands. So, essentially we're getting all of these little tiny fragments that are being made. And those little fragments being made are called Okazaki fragments. Named after the last name of the scientist who determined this. Their last name was Okazaki. It was a husband and a wife. Okay, so those Okazaki fragments are these individual pieces of DNA that are being built on the lagging strand. Because remember, helicase is opening this way, but polymerase is moving in the opposite direction. So we're opening up over here, but all of our enzymes needed for replication of the lagging strand, they're all over here on this side of the picture, right? So you're getting these little tiny fragments, and each of these fragments is going to start with an RNA primer. So primase has to prime here. It had primed here earlier. It primed here earlier. It primed here earlier. It had primed over here, but now we have our last enzyme to talk about, DNA ligase. And DNA ligase is the one that is going to come by and hook together our Okazaki fragments. So everywhere I've drawn a square is where DNA ligase is either currently functioning or is going to function soon. Okay, so DNA ligase is like a glue that puts together the Okazaki fragments. And my dogs are growling at each other. That's adorable. Okay, so that's all I have for you on this video.